We'd like to start so that we don't waste any of Dr. Sheffield's time. It's a pleasure to welcome you to the college seminar today and to welcome our special guest, the college's honored alumnus, Dr. Val Sheffield, who will be introduced in a few minutes. We've asked Dr. Will Winder, the chair of Physiology and Developmental Biology, to offer the invocation.
Our Father in heaven, we're grateful for this opportunity to gather together this day. We're grateful for the many blessings that we enjoy here in this college at Brigham Young University. We're grateful for thy Son and for all that he has done for us. We're thankful for the gospel that has been restored into the priesthood. We are grateful for the opportunity this day to honor Dr. Sheffield, one of the alumni from this college. We are grateful for his accomplishments and for his exemplifying the motto of entering to learn and going forth to serve. We are mindful this day of all of our alumni who are serving throughout the world and are making a difference. We're grateful that we can be here to learn and pray that thy Spirit will be with us in the name of Jesus Christ.
Amen. As an alumnus of the college, Dr. Sheffield has many friends here, and we welcome those who are also here visiting today. At this time, we'd like to ask one of his close friends, Dr. Koyman, to give us an introduction for Dr. Sheffield.
Val C. Sheffield received his bachelor's and master's degrees at Brigham Young University. After earning a PhD and MD from the University of Chicago and postgraduate training in pediatrics and medical genetics from the University of California, San Francisco, Val became a faculty member of the University of Iowa College of Medicine, where he has developed a highly successful human genetics research program. His laboratory is recognized worldwide for its work in identifying genes involved in hereditary blindness with the goal of prevention and treatment of vision loss. His laboratory has also identified novel genetic pathways involved in human obesity and birth defects. He is currently also an investigator of the prestigious Howard Hughes Medical Institute and director of the Division of Medical Genetics. He has over 250 peer-reviewed publications, many in prestigious journals such as Science, Cell, and Nature Genetics. Val and his wife, Kristen, also a BYU graduate, are the parents of 6 children. He currently serves as bishop of his home ward in Iowa City, Iowa. On a personal note, I would just say that Val is one of the nicest human beings I know and such a great example of what it means to be a BYU graduate. Before you speak today, Val, Dean Brown has a special presentation to make.
Val, we really appreciate you coming back to share with us. You really do represent someone who's gone forth to serve. And to help you remember today, you can lift the napkin if you want. We have something for you to take with you. This sculpture was done by Dan Fairbanks, one of our former faculty members who I think you also know. Awesome. We look forward to hearing you. Thank you.
This is all pretty embarrassing. But I'm glad to be back. When the dean called me to tell me that I had gotten this reward, I was pretty nervous about it and nervous about what to talk about. So I called up David Koyman and asked him what I should talk about. And he said, well, frankly, he wasn't very helpful. He said, talk about anything you wanted to talk about. So I came up with this. So I'm going to talk about some lessons that I learned at BYU, and I'm going to talk about some basic genetic principles. And then I'll talk about some interesting patients that have led to some of my research. And then I'll talk some science, and I'll give a summary. So that's sort of the overview of my talk. But when you get an award, I feel like you need to start out with some statements of humility and thanks. So I'll put these here. So Dean Brown called me up and told me I was getting this award, and so I called my wife and told her about it. And here's what she said: "Must be slim pickings in the actually, but one of the humbling things about this is I know it's not slim pickings in the nominee department, that there's lots of qualified people and more qualified than me. So that's the humility part. Also have the thanks part. Of course, I thank my family, BYU in general, and professors that have helped me over the years. And a few stand out in that I will be mentioning a few of these during my talk, but particularly Bob Sigmiller was my research mentor for my master's degree, and he was a terrific role model. Dwayne Jeffrey and Bill Bradshaw I learned lots from. And my good friend David Koyman is one of the greatest people I know. He's a man of great knowledge and great faith, and he's taught me a lot about faith in our time together. In 1970, I took a course, Zoology 101, I think it's called. It was taught by Professor Jim Barnes, and I was really interested in this subject, and I went to the course, and it was an outstanding course. And this is a picture of Professor Barnes. The dress code at BYU has changed. This obviously is Gregor Mendel, and in this course at BYU was the first I heard about Gregor Mendel, believe it or not. He was a freshman at BYU, and his experiments with pea plants. And I was very fascinated by this. Mendel studied 7 traits in peas. And each of these, and so here was the experiment. Mendel could take peas of different varieties with different traits and cross them together here. So here shows he's crossing, for example, a yellow pea with a green pea. And then the offspring of that, or the progeny from that, are all yellow peas. And then he crosses yellow peas with yellow peas from this first cross, from this generation here. And he gets back in the predicted combination only yellow peas that are either yellow or green. And he comes up from that with some very interesting laws or principles. The first law is the law of unit inheritance, where he recognized that characteristics of the parents did not blend in the offspring, but they sort of bred true, yellow or green, for example. He didn't get light green. If you think about it, there's an experiment that's been going on for eternities on that. If you cross a male with a female, you don't get something in between. You either get a male or you get a female, at least not usually. So with this idea led to the concept of genes. Then he also came up with the idea that these genes were in pairs, but only one member of a given pair is passed on from each parent to the offspring. So traits are inherited in a predictable ratio due to the inheritance of these pairs. And this is called the law of segregation. And then the third law is the law of independent assortment. And that idea is here. If you cross, say, a round yellow pea with a green wrinkled pea, in the second generation, you get some mixture. You get some wrinkled green ones, some wrinkled yellow ones, some round green ones. And so the wrinkledness and the color are segregating independently. And so that's called the law of independent assortment. So I was so enamored by this that when I got back from my mission, I decided to take more genetics courses, and I took general genetics. And here I signed up for a course by Professor Jim Farmer. And the first thing I recognized from that course was that it was too hard. And so I better drop out from that course. And I actually did drop out. Although I sat in all the lectures, I didn't take it for credit. And I don't know why, because I was very interested in it. And the course was offered again in the summer term from a different professor, Professor Dwayne Jeffrey. And I decided to stick it out this time and take the course from Dr. Jeffrey. And I figured I would do well in that course until we had the midterm. And at the midterm, I got 28% on the midterm. Okay, that was the bad news. The good news was 28% was an A. So I had the second highest grade in the class. The average on the midterm was 16, and 4 students in a class of 40 or something got zeros. So this is to let you know how hard BYU was back in the day. Okay. Well, after doing so well in that course, I decided to take more courses from Professor Jeffrey. So in 1973, I took human genetics. And there I learned that Mendel's laws applied to humans. And I also learned that there were exceptions to Mendel's laws. And I also learned that there's 2 types of human diseases. So and human traits. There's those types that follow Mendel's laws, and then the non-Mendelian ones that are more complex, that don't give the simple segregation according to Mendel's principles. And so this got me interested in Mendelian diseases. Mendelian diseases are caused by mutations in individual genes that have a large effect. There's approximately 10,000 different Mendelian diseases. Most are individually rare, but collectively they're common. They say about 10% of children have some Mendelian disorder. The Mendelian patterns of inheritance in humans are the same as in other species. They're dominant or recessive. And the human disorders are cataloged in what's called Mendelian Inheritance in Man, which used to be a published volume that was this thick and got thicker and thicker, and then it was 2 volumes. Now it's online Mendelian Inheritance in Man with the URL given there. This just shows back when I was a student here, in the early 70s, there were less than 2,000 known Mendelian diseases, and they've been cataloged so that there's many more now. Okay, I mentioned exceptions to Mendelian diseases. So in one of these classes, I learned about Thomas Hunt Morgan, who studied fruit flies. And what Morgan discovered was that some traits do not segregate independently. They're exceptions to the law of independent assortment. And so just an example of that here. So this is a human pedigree with a human dominant disease that causes blindness. And the circles are females, the squares are males, the open symbols mean they're not affected, the closed symbols means they are affected. So you see in these last 2 generations, you have some affected people, some unaffected people. And so if the gene that causes this disease were in chromosome 1 over in this area, and you were to look at some other trait that was over in this area, indicated by the red here, those would sort independently. Okay, and so they would adhere to the law of independent assortment. However, if you had some trait indicated by the blue that was near the gene for the disease, then they would not sort independently. And so you see that here, whenever you have this blue, you have an affected. Wherever you don't have the blue, you have an unaffected. And so these things are segregating together. And instead of this being a trait, you can have these, you can look at genetic markers that are polymorphic between individuals. And so one thing I did early in my career is I did something called genetic linkage mapping, using genetic markers that we had developed in our laboratory that were scattered across the genome. So you can use these genetic markers to look for markers that segregate in the same pattern as the disease, and that tells you where the gene is causing that disease. That's called genetic linkage mapping. Okay, so here's a quote I like. It says, "When I was young, I found out that the big toe always ended up making a hole in the sock," by Albert Einstein. He goes on to say his solution to this was to stop wearing socks. That's a simple solution. And the medical equivalent of this, finding a simple solution, is this quote by William Harvey, who was the father of the circulatory system. Harvey said, "There is no better way to advance the proper practice of medicine than to give our minds to discovery the usual law of nature by careful investigation of rare forms of disease." So in 1992, when I was a new assistant professor at the University of Iowa, I saw a patient in my medical genetics clinic. He was an 11-year-old boy. He came in, brought in by his mother, and the complaint was that he was going blind because of a disorder called retinitis pigmentosa, where he was losing his vision. He was also very overweight, obese. He had learning disabilities, and he had been born with something called polydactyly, where he had extra digits on his feet and hands. Okay, and they had, of course, been surgically removed. So I could go to this Mendelian Inheritance of Man and discover that this patient had something called Barney Beetle syndrome, or BBS. And the features of this disorder are the blindness, the obesity. These patients also get high blood pressure and diabetes, disorders that are very common that don't follow Mendelian principles. The learning deficits, the polydactyly, they also can have heart and kidney defects. So I thought this would be a nice disorder to study. The thing that is most devastating to the patients and their families themselves is the blindness. This is a friend of mine, Leo Hauser. Leo does not have Barney Beetle syndrome, but he does have another form of retinitis pigmentosa. And he is going blind. So this picture of Leo, this is what it would look like to Leo himself. Okay, Leo's a very interesting person. He's a multi-millionaire, highly successful toy manufacturer, but he is going blind, and he doesn't like the fact that he's going blind, but he's a very optimistic person, and despite having millions and millions of dollars, he cannot prevent himself from going blind. So, believe it or not, Barney-Beetle syndrome, which is a rare disorder, is a Mendelian disorder. It segregates as a recessive disorder following Mendelian principles. So, something else I learned at BYU was about this person, Sir Archibald Garrett. Garrett was an English physician who became very interested in a particular question, and this is that question. The question for Garrett was, why are some children born with abnormalities? And this wasn't a new question. In fact, it was quite an old question. From this scripture, and as Jesus passed by, he saw a man which was blind from his birth. So then his disciples asked the same question. They said, Master, who did sin, this man or his parents, that he was born blind? So he's asking the same question. These disciples are asking essentially the same question that Garrett asked. The Master's answer was, neither has this man sinned nor his parents, but the works of God should be made manifest in him. Garrett's answer was a little different. He said, we must seek an explanation in some peculiarity of the parents. So I'm sure you say the same thing about your parents, right? There's something peculiar about them, and that's why I am the way I am. But Garrett said there's some peculiarity in the parents which may remain latent for generations, but has the best chance of asserting itself in the offspring of the union of two members of a family in which it is transmitted. So what Garrett recognized, because he had just come to learn of Mendel's principles, was that some families are like this shown here, where the parents are actually related to themselves, related to each other in ways other than marriage. So in this case, they share grandparents. And in such cases, if a grandparent has a mutant gene, that grandparent can pass it down both sides of the pedigree until the offspring of the parent get two copies of that mutation, that recessive mutation, and this child has the disease. So this was Garrett's idea. And so, armed with this knowledge, I decided, well, we should study Barney-Beetle syndrome, but we don't have a lot of inbred populations in Iowa. So we'll go to southern Israel. And why would we go to southern Israel? Is that in southern Israel, they have Bedouin populations where the usual marriage is to marry a relative. So these marriages are arranged, and often it's a marriage between first cousins, sometimes between uncles and nieces. Okay, and so armed with this information, we actually had a University of Iowa truck that we bought over in Israel. Have a collaborator here who's a Bedouin Arab physician, Khalil El-Mador. Fantastic story in himself is Khalil's story. But Khalil would drive out in the desert and collect DNA samples and information on parents. and family history data on parents, and he was able to come up with Barney Beetle syndrome pedigrees that looked like this. So large pedigrees, multiple affected individuals, lots of inbreeding. And he didn't come up with just one pedigree, he found 3 pedigrees like this. And so we started to do genetic mapping of these pedigrees using DNA markers produced in my laboratory. And we had a surprise. In each of the different families, the gene causing this disorder mapped to a different chromosome. And so what did that tell us? It told us that really there were 3 different genes following Mendelian principles that each independently could cause this very same disease. This slide shows about 15 years worth of work by my lab and other labs. My lab's work in yellow, where the what's called heterogeneity of this disease was made even more manifest. Now there's at least 12 genes that can independently cause this same disease. Okay, so now we get to a little more science part of the talk. We wanted to figure out what these genes and the proteins coded by these different genes do. Just by looking at the structure of the proteins and things, we didn't really have a good idea, but we could come up with a couple of hypotheses. One hypothesis was that these different proteins coded by these different genes formed to make some molecular complex, a molecular machine, if you will, that had a function, such as if then one piece of that machine isn't functional, the whole machine doesn't function. And so there would be many different ways that you could break that machine. And we also knew that this must be important for a fundamental biological process, because it just didn't cause blindness, it causes other types of birth defects and other problems like the obesity. So we, to make a long story short, we did the experiments to prove that these proteins interacted together and formed a complex called the BBSome. And we could do further experiments. The BBSome consisted of 7 of the BBS proteins, but that means there were others to be found. So we did experiments to find if the others formed a complex, and indeed the others also formed a complex, or at least 3 of the others formed a complex. And an experiment shown here, which I won't go into detail on, we demonstrated that the second complex is needed to manufacture for the first complex to be assembled. And so that ties all of these different protein complexes together. Okay, but it still doesn't tell us really what these things do. And so we decided, well, we'll make some mouse models, and using homologous recombination principles, we can knock out the function in given mice. And so we made several mouse models by knocking out different of the BBS genes. And one thing we noted right away is that the mice went blind, and they went blind because their photoreceptor cell layers died over time. So here is a normal control, and you can see this layer here is gone in this mutant animal. And photoreceptors are found at the back of the eye. They're specialized cells that are shown here, rods and cones you've heard about, and that these rods and cones die because of the abnormal BBSome. But why do they die? And just to tell you, there's a hint here. These rods and cones have a special function. They have a cell body here that makes all its proteins that are transported through what's called a cilium to an outer part of the cell. And this transport mechanism is very important, and that's where we think BBS proteins play a role. Okay, a little bit about the physiology of the mice. So if we knock out one of these genes, instead of having a mouse like this, we get a fat mouse like this, just like in the humans. So we wanted to study the obesity in these mice. And so the first thing we did was we measured how much they ate and how much they move around. So the red and green are different animals, knockout animals, and the white is the controls. You can see the knockouts eat more and move around less than the controls. So they get fat for the same reason you and I get fat. If we do, we eat too much and we don't exercise enough. So, so we spent millions of dollars figuring out what you think we should have known, which my wife would say, duh. I could have told you that without doing an experiment. But we can learn more about this by knowing about a pathway that is known that fat stores produce a protein called leptin that goes to the brain and signals specific pathways in the brain to increase energy expenditure and decrease food intake. And if you look at the BBS mice, so leptin suppresses appetite. So you say, well, maybe the BBS mice don't have enough leptin. Well, that's not true. They have too much leptin, okay? Or they have plenty of leptin, because they have lots of fat-producing leptin. So they have high leptin levels. So they're what you call leptin resistant. And we can show that by an experiment. If you give a control mouse leptin, they stop eating. If you give a BBS mouse leptin, they keep eating, and they don't lose any weight. Okay, and that's because there's a specific receptor called the leptin receptor that binds to, and leptin binds to that receptor, and then it turns on a downstream signaling pathway through another molecule called STAT3, which becomes phosphorylated and then sends signals to stop eating. And so in the case of BBS mice, the leptin can bind to the receptor supposedly, but this STAT3 pathway is not activated, and we can show that by giving a wild type mouse leptin, they phosphorylate STAT3, where BBS mice have greatly attenuated phosphorylation of STAT3, so they're not turning on the signals. And what does this have to do with the BBSome? We've demonstrated in this experiment, which again I won't go into detail about, but that BBS1 binds directly to the leptin receptor. If you mutate BBS1, which is part of the BBSome, that binding is greatly diminished. So the BBSome plays a role in leptin receptor trafficking. That's our hypothesis at this point. Okay, so here's put together as a working model that the BBSome is assembled from these different BBS protein complexes with the aid of another complex, and that this BBSome aids in trafficking of vesicles and proteins to the membrane and perhaps to cilia. And one of these membrane proteins that's trafficked is the leptin receptor. Okay, now I'm going to tell you a little bit different story. This is a map of the Eastern Caroline Islands of Micronesia. And in one of these islands there is the Pingala Atoll. Okay, an atoll is a coral island with a lagoon in the middle. Okay, and I guess it's a pretty place. And around 1775, there was a typhoon, Typhoon Linkaeki, that hit the island, okay, and killed most of the population. The population that wasn't killed by the typhoon, the people, a lot of the people died of starvation, okay, except there were a few survivors, including the king. So I guess the king and his spouse or spouses, I'm not sure what it was, survived. And they repopulated the island. So that sounds a bit like the Bedouins, right, where they'd have quite a bit of inbreeding. And today there's about 3,000 people, and most of them can trace their ancestry directly to the king. And about 10% of the population, which is a high number to have a specific Mendelian disease, have a specific disease called achromatopsia. And what is achromatopsia? It actually means total color blindness. So instead of seeing this parrot like this, they see it like this. In fact, it's a bit worse than that because they are highly photophobic. So light is blinding to them. So in the daylight, they can't function, have to wear real dark sunglasses, and their visual acuity would be really bad. So at least in my way of thinking, this disorder isn't good. In 1969, Irene Momini, who's an ophthalmologist at Johns Hopkins, went to this island and collected blood samples from these patients and characterized their disease. Okay. In the meantime, we went to Israel. This is my daughter here with her Bedouin Arab, friends here was a bit, they liked her a whole lot. She's sitting in the audience here. So this is Abby and her friends. So we went there and we collected samples from patients with achromatopsia in the Bedouin populations. And to not go through the details of the work, we were able to use genetic linking mapping to map the disease to chromosome 2 in the Bedouin populations, whereas the Hopkins group mapped it to chromosome 8 in the Pingalopees Islanders. So another case of genetic heterogeneity. You see here we did it first. If you're not a bit competitive, you don't succeed. But at any rate, it turns out that the genes that cause these two different diseases produce proteins that form a complex. And so each of the two proteins are needed to work together to form an ion channel that, when not functioning, causes this complete color blindness. This has been chronicled, at least the color blindness part, as in the book by The Island of the Colorblind by Oliver Sacks. And so a quote from that book from someone with total color blindness says, "We don't just go by color. We look, we feel, we smell, we know, we take everything into consideration, and you just take color." So they're claiming that this disorder for them is a culture a little bit. And is this something we would really want to treat and do away with? In fact, they claim that they can see constellations in the heavens a little bit different and better than we do, because they have good night vision. And this is just a picture that might illustrate that. All right, so now I'm going to conclude with a few minutes on treatment. Okay, so Christ, going on with this scripture I gave before, where the disciples ask, why is this man born blind? And Christ said, so that the works of God can be made manifest. He goes on and says, and the rest of the story goes, as long as I'm in the world, I am the light of the world. When he had thus spoken, he spat on the ground and made clay, and anointed the eyes of the blind man with the clay, and said unto him, go wash in the pool of Siloam. He went his way therefore and washed, and came seeing. Okay, so this is pretty good. I want to know how Christ did this. What's in the clay? What's in the pool of Siloam? How did this happen? Well, in thinking about that, I continually think about my patients. And this first Barney Beetle syndrome patient I told you about that I saw some 18 years ago, he's now a 29-year-old male. At that time, we didn't have any genes that cause the disease. Now we have 14 genes that we know can cause this disease. We know he has a specific mutation in the BBS1 gene. Okay. He's currently blind. He's morbidly obese, despite our efforts to get him to diet. He continues to gain weight. He now has high blood pressure. This last time I saw him a week ago, he had developed diabetes, and he has high cholesterol. He's not retarded. He does have some learning disabilities. I helped him get a job, so he's employed. He works at P.F. Chang's in Nashville, Tennessee, and he helps roll the silverware and the napkins to prepare for the diners. The extra fingers and toes haven't grown back, which isn't surprising. But what have we done for him? What can we do for him is the question. So there are interventions. We can, since we know genes, we can diagnose carriers. So this is something we do in the Bedouin populations where the marriages are arranged. If the person arranging the marriage says, should this girl marry this guy? Should I arrange this marriage? We could tell them, yeah, that would be good because neither are carriers, or one's a carrier, one's not. We wouldn't say that. We'd say, yeah, that's a good marriage. But in other cases, we could tell them to avoid certain marital arrangements, and we've been asked that question on a few occasions. We can diagnose specific forms of blindness by knowing the specific mutation, which allows us to predict specific prognoses for certain mutations. And the goal is to have specific treatments, pharmaceutical treatments, and perhaps gene therapy. So one of the treatment was very simple. So a case where we made a specific diagnosis, the BBS3 patients, it turns out, have very mild retinitis pigmentosa. So their photoreceptors live. But people didn't know this and were treating them as if they were blind because they're very nearsighted, when all they really needed was someone to bring them glasses. So we arranged for glasses to be sent over to the desert in southern Israel and treat these patients that way. So that was a simple treatment by making a specific genetic diagnosis. Okay. In 1953, Matthew Cowley, who was then a member of the Quorum of the 12 Apostles, told the following story at a BYU devotional. He said, I've told the story about the little baby 9 months old who was born blind. The father came up with him one Sunday and said, our baby hasn't been blessed yet. we'd like you to do it. I was just about to start when he said, by the way, give him your vision when you give him a name. He was born blind. It shocked me, but I had faith in the Father's faith. After I gave the child its name, I finally got around to giving it its vision. Okay, so now can we give anyone with science their vision? So one thing we do at the University of Iowa is we have a center where we diagnose specific forms of blindness genetically. And then we're collaborating with people at University of Pennsylvania to do gene therapy on a disorder called LCA. It stands for Leber's Congenital Amaurosis. That's why we call it LCA. So I'm going to show you an 8-year-old boy who had very poor vision since birth, essentially born blind. The retina is intact, so he has a normal eye, it just doesn't work. And here is this boy prior to treatment. And he's walking through, he's supposed to follow arrows on the floor and walk through a sort of maze. You don't really need to hear what he says. He says, this is hard. This is being really hard. And then he just freezes here. And the investigator says, you don't see any lines on the floor? No, I don't. I can't even see anything. Okay. So then this patient was treated 90 days, well, he was treated with a single subretinal injection with a virus carrying a gene, the RP65 gene, that is functional. And this virus targets the retina. And so this is 90 days later. He steps over something, falls down.
Wow. Yeah, next door should be bigger, you're right. I think it should be.
Okay, so in summary, if you're studying something complex, it's good to simplify it. The study of rare disorders can lead to some great discoveries and some really basic science insights. We've discovered a new molecular complex, the BBSome, which plays a role in intracellular trafficking. And we've investigated some new human disease insights, including a new mechanism of leptin resistance leading to obesity. And we're pursuing interventions, which I think will be very successful. The end of the story about from Matthew Calley in the BYU devotional goes as follows. That boy is almost 12 years old now, so this was 12 in 1953. The last time I was back there, I was afraid to inquire about him. I was sure he'd gone blind again. That's the way my faith works sometimes. So I asked the branch president about him. He said, Brother Callie, the worst things you ever did was to bless that child to receive his vision. He's always getting into mischief. I was thrilled with that kid always getting into mischief. And in the same talk, he made the following quote: medical science had laid the burden down and God had picked it up. So a final summary point is that medicine can do a lot. And medicine and science and medicine and God can go hand in hand. Many people in my lab and many collaborators. And I'd like to specifically acknowledge these two, Khalil El-Bador and Rivka Karmi. This is a collaboration between a Bedouin Muslim, an Israeli Jew, and an American Mormon, which in itself is maybe another miracle.
I keep telling them, I said, guys, that is not going to happen. It's sort of wishful thinking, because I said, you need to prepare for this emerging giant that's in the neighborhood, because it is the 800-pound gorilla. But that is a huge, huge concern that they have.
So net bottom line, is the double-digit growth sustainable?
Not without, adjustments along the way. No. No, I mean, there's going to be bubbles and that kind of thing. Yeah.
Thank you so much for being here. I'm wondering if you'd be willing to share any personal experiences you had in your time as Vice President. It would be either constructive, entertaining, or harrowing.
Or harrowing. Oh, harrowing.
The one thing I'll tell you, the difference between politics and business. In business, I always sort of, you have a buyer and a seller, and I've always sort of made the equation that's like Republicans and Democrats, okay? You argue, you bicker, sometimes you get a deal and sometimes you don't get a deal. And in the business world, once the buyer and seller, they come together and they sign the contract, they all go out to execute the contract. Both parties, seller and the buyer. In the world of politics, where I was for a number of years, here's the way it worked. You had the House and you had the Senate. And you had Republicans and you had Democrats. And you would sit there and you would argue and you would bicker, and it'd pass the House, and then it's sent to the Senate, and the Senate would send it back to the House, and the House would call for a conference, and you'd go to conference, and you'd have Republicans on one side and Democrats on another side, and you'd argue and you argue, and sometimes it comes out of conference committee, goes back to the two houses, and it finally gets to the president. And these things take, months to get there, sort of like a deal, You strike a deal and you got the due diligence and everything. It takes months to sign it, right? But once it's signed in the business world, it's done. You go down there and you have this huge signing ceremony with the president. He puts his signature on the legislation, and he's got the speaker and the majority leader and minority leader and cabinet members all behind him, members of Congress, and he signs this thing. He says, this is it. Now let's go out and execute. Those people all leave that room and they go out and try to undo what they just agreed to. Now, that's the difference between politics and the world of business. But no, the vice presidency is you're president of the Senate. You're paid by the Senate. You're not paid as an official member of the executive branch. Richard Nixon was the first vice president who moved his main office from this U.S. Senate down to the old executive office building in 1952.
Yeah, I think so.
And then Walter Mondale in 76, or actually 77, created a vice president's office in the West Wing. where it remains today. You have an office. You actually have a lot of offices as vice president. You have one that's in the West Wing, which is right very close to the Oval Office. You have one in the old Executive Office building, which is across the way, and then you have a couple offices on Capitol Hill. In the Senate, you have one, right next to the Senate, and you have one in one of the Senate office buildings, and then you get one in your home state. So, I mean, on a boring day, you just visit your offices. This being vice president, But it's, everyone they sort of laugh at the vice presidency because it's, over the years, I mean, it's a fairly complicated office. The office itself and what a vice president does is really determined by the president. It's the relationship that you have with the president of the United States. In my particular case, I was very fortunate because George Bush had been vice president for 8 years under Ronald Reagan. So he knew the ups and downs of being vice president and was very sensitive. And where vice presidents can really get cut out is at the staff level. I mean, any vice president, unless they just don't speak, Humphrey and Johnson were probably an exception to this because they didn't really speak that much, and Lyndon Johnson didn't even let Hubert Humphrey have the vice presidential desk because they're all the vice presidents we have all signed in there, and there's one missing. It's Hubert Humphrey going all the way back to Truman. And the reason is because Johnson didn't want to give it to him, and he took it when he's president. But with the exception of that, all vice presidents, they can walk into the Oval Office and talk to the president anytime. But where you get cut out is at the staff level, and because the staff really does run things a lot. An inordinate amount of power of the staff, and that's why it's very interesting now in the Obama administration. At least I'm interested in it. I think you would be too, as you try to read the tea leaves and what it means with Larry Summers leaving, Christina Romer left, is Geithner next. I mean, my reading of it is that they are really setting the stage to run a very political operation from the White House these next 2 years. And therefore, the economic policy is going to be probably a lot more economic populism. It's going to be things that are going to play well in Peoria, as they say, and on the campaign trail, rather than substantive economic prescription that is necessary for us to get that, that darn unemployment from 9.5% down. So the staff is very, very important. But the office is, not to digress, but the office of vice president, it is really determined by the president. And I've always said there's 2 requirements of being vice president. One, to be prepared, and you have to. And I think all the vice presidents, most of them before, well, I'm sure Mondale did, was certainly included. I don't know, in the Nixon years, and also in the Ford. And Rockefeller was actually cut out by the Ford staff when he was vice president, to Jerry Ford. But Gore, Cheney, and Biden, they all had the same access to information that I did. And it's very important that you have total access to all the information that the president has. So that's one, is to be prepared. And 2, from my point of view, is to be loyal. It's not your agenda. Different administrations operate differently. Sometimes they think it's okay for the vice president to disagree with the president. I never, in cabinet meetings or anytime we had a meeting more than 8 people and any meeting outside the Oval Office with the president around, I would never, ever differ from him, even though I would in private, just because people would say, okay, well, the president and vice president are at odds on this thing, and it would get in the paper. And George Bush, he didn't do that when he was vice president. You didn't read any disagreement with Ronald Reagan when his 8 years, because he was totally loyal, and you didn't read any disagreements that I had with President Bush. Although we did have, obviously you're going to have some disagreements. They're never huge. There was never a philosophical gap, but it really does come down to what the president wants. Yeah.
Moving back to Ben for a second. On the personal side of studying the years, I'm curious, what is it they actually believe in? What motivates them? Why do they do what they do now if they've so veered away from the
Oh, I'm sure they do. They will tell you that they actually have a democratic system, because the Communist Party, they elect their local leaders, and the local leaders elect their municipal leaders, and the municipal leaders elect their provincial leaders, and the provincial leaders have a say in the People's Party Congress that meets every 2 years, or I think it's every 2 years, or maybe, I think it's every 2 years. And they will tell you point blank that they don't know this is democracy. And then you go, okay, well, fine. Now let's talk about what a real democracy is. And what they will say, they say, we're moving in that direction. They say that. And then they'll say, it takes time in China. And they always like to talk about Hong Kong. Remember Hong Kong, what was it, 100 years that the UK had it? Something like that. It was a long, long time.
99-year lease.
99-year lease. You know what? They waited. 99 years, no big deal. They've been around. They go back all these dynasties and everything. They centuries that they have. So 99 years, that's not a big deal. So they will tell you that. Now, look, they understand that the young people today over there laugh at the Communist Party. They know that outside the borders, they keep referring to it as the Communist Party. You don't hear them talk about the Communist Party. They talk about the Chinese People Party. I mean, it is a communist system, but there's not a lot of room for dissent, but some. I mean, they do try to listen to what's going on. They do make changes. It's not a totalitarian regime like Tehran or when Saddam was there, or the way Castro runs Cuba, but it is an authoritarian regime, and they can be very, very tough at times. But no, they will point blank tell you, look you right in the eyes, and say we're moving in that direction, and they just say it may take some time. So, but it's going to continue. They think it's very efficient. And if you want to look at how quickly they have risen in the last 2 decades, it's hard to argue with them. Now, you got to look forward and say, well, that's not the best thing for your people, because your people aren't really free. In the long run, that's not going to be the best, because you're not open. But they're saying, look what we've done. They'll argue, some will argue, and they're having this discussion. I'm sure you're having it right here in some of your political science classes. What's the best form of government these days as far as efficiency of creating jobs, creating wealth, maintaining the peace? I mean, we all believe, and I firmly believe, that it's democracy, that it's the kind of democracy. It may be messy like we have, but it's the best form of government. But